Hello, this is a video for functional anatomy and we are looking at topics in the muscular system. So this is the second of two videos looking at the neuromuscular process of muscle contraction, how our nerves and muscles work together to make muscles contract. The first video looked at more the nervous side of things, how we send an action potential along a neuron to innervate a muscle fibre. And this video looks at the process with which actually the components of muscles work to make contraction happen and, typically speaking, shorten. And this is called sliding filament theory. Here's the content from the specification. So you can see there's quite a lot of language, names of parts and a little bit of process for this theory. It's just something that will come with familiarity. So you will get used to the language and the terms. Some things to start off then. In terms of the anatomy, this is our motor neuron. This is our neuromuscular junction where the nerve meets the muscle. These vertical bits here that look like shoots are actually T-tubules or transverse tubules. Now, when the electrical impulse arrives at the muscle fibre, it will spread and it will spread down the T-tubules and get to these yellowy blobs here, these stores of calcium. And the stores of calcium have a really difficult name to remember. Here's a challenge for you. The store of calcium is called the sarcoplasmic reticulum. So really all this is giving you is a little bit of anatomy that you'll need to know in terms of how you explain the sliding filament theory. You'll need to know what T-tubules are, and these are the tubes that the action potential spreads down, and you'll need to know what the sarcoplasmic reticulum is, and it is basically the store of calcium that the action potential gets to, and the calcium is released from that store. And the calcium being released from that store, so it looks like it's represented as triangles in this diagram, that calcium is the starting point of the sliding filament theory story. So the impulse, the action potential comes down a neuron, it's transmitted across the synapse by ACH. If enough gets across, the action potential will spread down the T-tubules and it will trigger the release of calcium from the sarcoplasmic reticulum. So again, before we actually get onto the process of the sliding filament theory, I'm giving you some context, some anatomy. You should recognise this sort of breakdown of the whole muscle, the fascicles, the muscle bundles. This is a single muscle fibre, which is made up of lots of myofibrils. And the myofibrils look striated or striped. And the reason they look striated or striped is because they are made up of lots of these units. Now, one of these units is called a sarcomere. So you can just about see the dark and light sections of that myofibril. That's these dark and light sections here. This is a light bit, this is a dark bit, this is a light bit, this is a dark bit. This is one sarcomere unit. And a sarcomere is the contractile unit of a muscle fibre or of a myofibril more specifically. And you can see here there's 3 in a row, but all along a myofibril there'll be lots and lots and lots and lots of these. And ultimately what's happened is these work a bit like a concertina. These green strands are pulled towards the M line, pulled towards the middle, and they overlap. They slide past the red lines. That's pretty much what sliding filament theory is. But you need to know what is a sarcomere. It's this unit here. You'll need to be able to label any of these bits. So you can see, for example, the M line is like the middle line. It's the middle of the sarcomere. The ends of the sarcomere are Z lines. Now, normally when I draw it in several diagrams, it's almost zigzag, which is a big clue for them being Z lines. One component of a sarcomere that doesn't ever change, it stays the same length the whole time, is this A band. So you can see it's the A band. The A band is always the length of the myosin myofilaments, the red in this diagram. Now that will always stay the same length. This H band, which is just red, may disappear, but it's the central bit here where it's just the myosin. And then there's a sort of a green, what we'd call a light band over here, which is the I band, where it's just actin. So this is the I band. Things to learn, again, familiarity will come with, practicing this terminology. Just to show you a slight, again, more microscopic version of the same, here is the sarcomere. It doesn't look exactly like the diagram, of course, but we've got the middle of the sarcomere. We've got the H band or H zone, sometimes it gets called, which is just myosin. We've got where there's actin and myosin. The A band is the length of the myosin arms. We've got the lighter bit, which is the I band. Now you can remember light with I. Light is the I band. Dark is the A band. And that's obviously the Z lines. Call them discs here, but you need to know Z lines. And this is a single sarcomere, and there's two myofibrils beside each other. This is a zoomed out version showing all the striations of myofibrils. Let's look in more detail then at a sarcomere, and of course this is a slightly more anatomical diagram here. So these were red in the previous diagram. This is the myosin with the arms and the head, the myosin blobs on the end. These are the sort of do all the work in sliding filament theory. You've got these globular looking actin myofilaments attached to the Z lines here at either end. So these are attached to the Z lines. They get pulled. So these do the pulling and these do get pulled. So just to identify the two different kinds of myofilament you'll need to know and talk about in this story. Actin are the thin myofilaments, so they are globular. The blobby ones here. Here's a better diagram. On this, though, there's more detail again. So there are special sites on the actin whereby these myosin heads need to attach to. So they're called actin binding sites. And they're hidden by tropomyosin, this swirly whirly protein wrapped around the actin. And the third thing you need to know is that troponin is attached to tropomyosin. So troponin is these blobs attached to the swirly tropomyosin, and at rest, the tropomyosin is covering the actin binding sites. So that's the globular ones there, actin. These ones with the arms and the heads, here's an example. Here is one of those arms. The myosin, let's call it a tail, I call it an arm, with the head on the end. And this is, of course, what wants to attach to the actin eventually. These myosin heads have ATP attached to them. ATP gives them energy to do all that pulling power. So you can pause the video and read the text in these blocks there for a bit more information. This diagram down the bottom left is actually a cross-sectional version of the myosin with the arms and the heads, and the actin, the thin globular ones like this. I talked about these a little bit earlier, but again, you can pause the video and take more time and care to look at the different labelling components of the sliding filament theory of a sarcomere. So here's our zigzag Z-line sarcomere. Here's a contracted version. Here's a relaxed version. And I'm hoping that you've noticed the change in shape. The fact that this is wider and this is pulled shorter. And that's what happened when all of the sarcomeres along a myofibril contract. The myosin arms pull the actin past them towards the M line, the middle, and that sarcomere shortens when a muscle contracts. So again, please pause the video, have a look at all these labels. You'll need to know what they are. You'll need to possibly have a diagram, and if there's an arrow or a number, a letter by one of them, you might need to identify what is the A band, what is the I band, what is the H zone, etc. So this was an exam question, an 8-marker exam question. So every paper, question 10, is worth 8 marks, and it's on any topic in the specification. So this one happened to focus on the sliding filament theory, and it wanted you to, it's given you some information, it's shown you a relaxed, long, widened sarcomere, and it's shown you a contracted sarcomere with the two types of myofilaments. It's given you nothing else. It wants you to analyse how the sliding filament theory accounts for muscle contraction in a sarcomere. So essentially, in detail, what happens to go from here to go to here in this contracted state. So I've used this mark scheme in my description of the process of the sliding filament theory. So this is the mark scheme on the right here, and I'm just going to refer in this slide to the first few red bits of factual information that was on the mark scheme. And actually, we've talked about this already. So what is the process of the sliding filament theory? Well, first of all, an action potential travels down a motor neuron, is transmitted across the synapse by ACH. The action potential goes down the T tubules and triggers the release of calcium from the sarcoplasmic reticulum, this store of calcium. And this all happens in the myofibril. And this is a single myofibril in the diagram. This is one sarcomere of the myofibril. There'll be another sarcomere up to the left and up to the right and many more along. So that basically is the first few points on the mark scheme. How does the impulse get from the neuromuscular junction to the myofibril? And it's about the T tubules and it's about calcium. Now this second slide is about these three bits of information now. The calcium's been released. What's the role of the calcium in the sliding filament theory and how does it help? Let's look down the bottom here. So in a relaxed state, can you see just under the tropomyosin, remember the tropomyosin is the swirly-whirly protein, there are blobs. There are little purple blobs that are hidden. You can see them this side. They're the binding sites. Now this tropomyosin is blocking those binding sites. And for contraction to happen, this myosin arm needs to attach to those purple sites. So what's the role of calcium? Well, calcium will bind with, so on this top diagram, calcium's this blob, will bind with troponin. I'm afraid the colours are different on different diagrams. It's quite frustrating. But the calcium in this top diagram will bind with these purple pinky blobs here. That's troponin. And what that does is it changes the shape of the troponin and pulls the tropomyosin swirly out the way. So down here, in fact, it's shown on the right-hand diagram, calcium binds with troponin, this yellowy on this diagram. It changes shape, and pulls the tropomyosin out of the way. Now, what that does is it reveals the actin binding sites, the myosin binding sites, they've called them. And you can see in this diagram now, one of the myosin heads is forming a cross bridge. That's what we call it when the myosin connects to a binding site. It's called a cross bridge. So that bit of the process is really simply stated over on the right-hand side here in the red. So then we need to know with, well, so what? What does this cross bridge have to do with anything? So the next thing that happens is what we call a power stroke. And this is almost like rowers rowing. These arms, these myosin arms and heads connect, unconnect, connect, unconnect. They will, they form a cross bridge, they break the cross bridge. But when they are formed and are connected, they essentially pull the actin and slide the actin past. Now remember, the actin is connected to Z lines, the end of the sarcomere. So this actin is connected to the Z lines. So if you pull the actin right to left, you're actually going to pull the Z line that way. This would be happening in reverse on the other end of the sarcomere, so that the Z line would be pulled left to right. So what happens then? We've got a cross bridge. The cross bridge is formed between the actin and the myosin. Great. So this diagram is showing that here. Then we need to use the energy from ATP to pull that myosin head, pull the actin. And that's the power stroke. That's called a power stroke. Then we need more ATP to arrive and attach to the myosin so that it can detach. If you think about swimming, perhaps you do one stroke with one arm, then you pull, and then you go again. It's repeated, almost like a swimming stroke. And that's what these myosin arms do. They are connecting, stopping, connecting, pulling, disconnecting, connecting, pulling, disconnecting, all at different places along the actin to pull it and to pull the Z lines towards the middle of the sarcomere. So key points here. We've got a cross bridge formed. ATP has energized that myosin head. It does the power stroke, pulling the actin and the Z line. And then we need more ATP to break down, to break that cross bridge so it can go again and again and again. Pause the video, look at both. These are two separate diagrams. They go through exactly that process. Take time, work through, get a sense of how the process works. I quite like this left-hand diagram. It's really simplistic, but it shows the blue people here are representing the myosin that do the pulling on the pink actin, which is moving the Z lines to the middle. That, again, this end of the sarcomere is pulling left to right. This end of the sarcomere is pulling right to left. And you can see how the Z lines get both pulled to shorten the sarcomere during contraction. And we're nearly at the end of the story. So we've got two versions of relaxed sarcomeres, our microscopic version where there's the Z line, there's quite a lot of I band at each end. There's the A band, which always stays the same anyway. There is an H zone in there that's got some lightness because it's just myosin. This is the middle of the sarcomere. This is actually showing two sarcomeres. So here's a relaxed sarcomere. There's an H zone in the middle. There's plenty of I band, so there's whiteness there. Once the Z lines are pulled by the myosin, by the actin, by being connected to the actin, note what happens to the I band. The I band was quite extensive here, so there's a big gap of just I band. Here it disappears. Here there was an A band, a white, sort of a lighter bit in the middle. It's shown better actually on this diagram. It's disappeared down there as well, so the A band disappears. The more overlapping and sliding over each other there is, the less I band and H zone there is. So the last bit of sliding filament theory is the Z lines are pulled closer together, which shortens the sarcomere. The H and I bands disappear. The A band always stays the same. There's just a different diagrammatic version of it. Just have a little look, pause the video. The left-hand side is during contraction, what happens, the process, which we've just talked through. But actually, the right-hand side is what happens in order to relax muscle. So let's say you've contracted and don't need contraction anymore. Kind of the reverse happens. So calcium is taken away. It's reabsorbed back into the sarcoplasmic reticulum. There's no more ACH transmitting impulses, and so on. The whole process is sort of reversed. Here's a very detailed version, so just pause, have a read through this, of how you make muscles contract. So this is a more elaborate version of the mark scheme. And here's a version of, again, how we end contraction to go back to a relaxed state. So questions we've had. There's been a 2 marker to identify these things in a sarcomere. Now that's quite a kind one, I think. Actin and the Z lines. It could be more complex. You could be asked for any of the bands or the zones. This was the 8 marker, and that, of course, is the mark scheme that I've been showing you.